Hello everyone, my name is Vimal and I am working as an assistant professor in IIMT College of Pharmacy, Greater Noida. And today I am going to present a very new topic, that is enzyme inhibitors in medicine and enzyme inhibitors in basic research. So this slide covers the following content, that is enzyme inhibition and enzyme inhibitors in medicine and enzyme inhibitors in the research. So before going to start this topic, we have to learn about the enzyme. What is enzyme? So enzyme is basically a biocatalyst. Enzyme is basically is a biocatalyst which is composed of various types of amino acid. So enzymes are soluble, colloidal, organic catalyst formed by living cells, specific in action, protein in nature, and inactive at 0 degrees Celsius and destroyed by moist heat at 100 degrees Celsius. So basically, enzymes are the soluble and colloidal organic catalyst which are formed by the living things. Living things means human beings or animals such as pigs and human. We synthesize the enzyme, various enzymes such as insulin, aldolase, and proteinase. So basically, these enzymes destroy uh, break down the certain types of chemicals such as protease breaks the proteins and insulin breaks the glucose. So these type of enzyme plays a vital role in management of the health of our human body and animals. So secondly, description. Enzyme are a specialized protein which catalyze various biochemical reaction. The concept of enzyme inhibition is routinely utilized to affect biosynthesis and metabolic pattern of various hormones, autocoid and neurotransmitters. So enzyme inhibition is a very new target for treating any disease. Suppose one enzyme is causing any particular disease, such as cancer. In the cancer, topoisomerase enzyme is used for the cell division. So if we block the topoisomerase enzyme, we can also block the cancer, or we can also cure the cancer. So secondly, how enzyme catalyze the reaction. Enzyme provide a reaction surface and suitable environment. Enzyme act as an surface for any substrate to start the reaction. That is, secondly, enzyme brings reactant together and position them correctly so that we can bond in the reactant, so that they easily attain their transition state and enzyme weaken bond in the reactant. So secondly, enzyme brings together the reactants and position them correctly so can the reactant fit into the enzyme and weaken the bond of the reactants so that they can react with another molecule. Secondly, thirdly, enzyme may participate in the reaction mechanism and enzyme form stronger interaction with the transition state than with the substrate or the product. So this pictorial graph demonstrate the how enzyme works. So in first picture, substrate entering the active site of the enzyme. Substrate entering the here. Substrate entering the active site of enzyme, and these are the substrate and this is the active site. So, and in the second picture, enzyme change shape slightly as substrate bind. Enzyme automatically change its shape to fit the substrate in it. And thirdly, enzyme product complex. Enzyme form a complex with the substrate. And in the last picture, the product will separate out and the substrate will separate out and the enzyme will also be separated out. So enzyme basically is used for the preparation of any product from the substrate by using the various substrates. So what is enzyme inhibition? Any drug or any molecule which blocks this mechanism or this reaction is called as enzyme inhibition in the simple way. And in the definition, enzyme inhibitors are molecules that reduce the catalytic activity of enzyme, reducing of effective enzymatic activity or complete blocking of enzyme may cause either complete death of cell, either modification in the pathway, such as in the treatment of cancer. We basically block certain enzymes, certain types of enzyme to destroy the cancerous cell, to block the cell division and to block the cell proliferation. So this is our main target. They are usually specific. Enzyme inhibitors are very specific and they work at the low concentration. Enzyme inhibitors are very specific, such as they can bind only to the specific target for which they have made. And thirdly, they block the enzyme, but they do not usually destroy it. They only blocks the enzyme but and not destroy the enzyme. ये आपका एंजाइम को डिस्ट्रॉय नहीं करते, ये बस इसको कुछ समय के लिए ब्लॉक करते हैं ताकि जो रिएक्शन है वो आपकी स्टॉप हो जाए. And then inhibition of biochemical pathway will be most effective if drug act on rate limiting step, the conversion of tyrosine to DOPA. Inhibition of biochemical pathway will be most effective. So enzyme inhibitors will be most effective when they used during the rate limiting step in the biochemical synthesis. So this is the classification of enzyme inhibitors. In the first graph, in the first picture, this is non-competitive inhibition, and second picture, competitive inhibition, and the third one is end product inhibition. In the first picture, we have seen that this is our enzyme, and this is inhibitor binding site. The enzyme have a different site on which the inhibitors bind, and one site is for the binding of the substrate binding site. And after that, when the substrate bind and no inhibition generated, the product will be such as without inhibitor of binding, the substrate can bind, causing the chemical reaction to be carried out and for the end product to be produced. When only substrate bind to the enzyme, we will have the products and product. And if the inhibitor bind on the inhibition side of enzyme, we will not get the product, such as substrate no longer fits to its binding shape because inhibitor bind at the enzyme and change the shape of the enzyme so that the substrate will cannot be bind to the active site of the enzyme, and at last we don't get the product. In competitive inhibition, the in substrate and inhibitors both have the same shape and same size. So when we give inhibitors, enzyme inhibitor to any patient or any human being or animal, inhibitors will compete with the substrate for the binding to the enzyme. And if the substrate bind, we will get the R and product. And if the inhibitor bind to the active site of the enzyme, we will not get our products. In the graph, we can see both the processes, such as this is enzymes, this is substrate binding site, and this is also the binding site for the inhibitors. When the substrate bind, no inhibition. If the substrate bind to the substrate binding site instead of inhibitor, the chemical reaction is carried out and the end product is produced. And when the inhibitor bind, if the inhibitor bind to the enzyme, the chemical reaction which would be carried out by the enzyme does not happen. The reaction is not carried out as no substrate can bind to the active site of the enzyme. Thirdly and lastly, end product inhibition. In this type of inhibition, the inhibitor work at the end point after the generation of any product. So here we can see the substrate binding site and allosteric site. Substrate bind for the chemical reaction. Chemical reaction will start and end product, we will get the end product. And end product inhibition occur when too much of end product is created, causing there to be an excess. The extra product will bind to the allosteric site, changing the shape of the substrate binding site so that substrate can no longer bind, stopping the metabolic pathway. In this type of inhibition, our product will act as a inhibitor. Product synthesize, and it will repeatedly bind to the allosteric side of the enzyme so that the end product will be of large number in a huge amount. So it will also block the uh, process of generation of end products and products, and it will stop. It will lastly stop the reaction, chemical reaction. Enzyme inhibitors in medicine. A selective inhibitor may block either a single enzyme or a group of enzymes. This will result in either a decrease in the concentration of enzymatic product or an increase in concentration of enzymatic substrate. The effectiveness of an enzyme inhibitor as a therapeutic agent will depend on the potency of inhibitor, its specificity, the choice of metabolic pathway, the inhibitor or derivative possessing appropriate pharmacokinetic characteristics. So, any enzyme inhibitor to be used as a medicine, these following characteristics should be in the enzyme inhibitor. The potency of inhibitor, its specificity means it specifically bind to the specific target enzyme, and the choice of metabolic pathway. By choosing metabolic pathway, we can choose our actions in the enzyme inhibitors. And its pharmacokinetic property. Pharmacokinetic property means uh, action of any enzyme inhibitor after entering the body, mainly the deposition and transportation. And enzyme inhibitor in medicine. Low doses and high specificity combined to reduce the toxicity problems. High specificity can avoid depletion of the inhibitor concentration in the host by non-specific pathway. Good bioavailability of the drug is also important. These are some example of the enzyme inhibitor. Enzyme inhibitor used in treatment of bacterial, fungal, viral, and parasitic disease. Clinical use, firstly, antibacterial. Enzyme inhibitor is dihydrofolate reductase, DHFR, and the drug which inhibit the enzyme are trimethoprim and methotrexate. And methotrexate is also used in the treatment of cancer. And secondly, antibacterial. Second enzyme is alanine racemase, and the drug which block the activity of this enzyme, and the drug which is used as enzyme inhibitor is D-cycloserine. And the third class is antifungal, in which the fungal squalene epoxidase is inhibited by the terbinafine and naftifin. And in the antiviral category, DNA and RNA polymerase, which are used for the generation and replication of the cells of viral cells, are the main target, such as DNA polymerase and RNA polymerase. And the drug which inhibit these uh, two enzymes is cytosine arabinoside. And in next category, antiviral, viral DNA polymerase enzyme is also inhibited by the acyclovir and vidarabine. And lastly, anti-protozoal ornithine decarboxylase is inhibited by using alpha difluoromethyl ornithine. And in hyperlipidemia means concentration of lipid will be increased in our body. So enzyme which reduce the concentration of lipids in our body is HMG coenzyme A reductase. So this enzyme will be inhibited by simvastatin, which which can reduce the hyperlipidemia condition of hyperlipidemia and lower the level of lipid. And lastly is antidepressant. Here the enzyme inhibited by the drug is monoamino oxidase, which is mainly responsible for the degradation of dopamine and norepinephrine and noradrenaline. So by inhibiting this particular enzyme, we can improve the condition, we can treat the depression. So the drug which inhibit this enzyme is phenelzine and cypropamine. And lastly, antihypertensive, the enzyme which is inhibited by the drug is ACE. ACE, full form of ACE is angiotensin converting enzyme, which is responsible for the high blood pressure, vasoconstriction. And the drug used for this to block this enzyme is captopril. And in gout, xanthine oxidase is inhibited by using the allopurinol. And in treatment of ulcer, ATPase enzyme is inhibited by omeprazole, which is proton pump inhibitor. Example of enzyme inhibitor used in the treatment of cancer. Type of cancer, enzyme inhibited and inhibitors. Benign, in the treatment of benign prostatic hyperplasia, steroid 5-alpha reductase is inhibited, which is inhibited by the finasteride. And in the second type of cancer, that is estrogen mediated breast cancer, aromatase enzyme is inhibited by glutathione. And in colorectal cancer, thymidylate synthase is inhibited by 5-fluorouracil. And in small cell lung cancer and non-Hodgkin lymphoma, topoisomerase second, which is used in a DNA replication, is inhibited by using the drug etoposide. And in hairy cell leukemia, adenosine deaminase is inhibited by the pentostatin. So, enzyme inhibitors in basic research. Enzyme inhibitors have found a multitude of uses. Number one, as useful tool for elucidation of structure and function of enzyme. Second, as a probe of chemical and kinetic processes and in the detection of short-lived reaction intermediate. Thirdly, for the product inhibition pattern, provide information about an enzyme kinetic mechanism and the order of substrate binding. And the fourth one is covalently binding enzyme inhibitor have been used to identify active site amino acid residue. Fifth one is reversible enzyme inhibitor are used to facilitate the enzyme purification. Sixth one is immobilized enzyme inhibitor can also be used to identify their intracellular target, whereas irreversible inhibitors can be used to localize the and quantify enzyme in vivo. And this is the reference from which I have covered the content of this slide. And then thank you.